So in this lecture, we're going to dig a little bit deeper into p-hacking because it's one of the biggest threats to statistical validity. So in part one, we learned that there are a lot of false positives, or potentially a lot of false positives. We don't know exactly how much, but it's worrying us now. And that this is probably because of something I'm calling p-hacking. So just as a reminder, in this 2x2 table, we, as researchers, we know which row we end up in, the top row or the bottom row, but we don't know which column we live in, the left column or the right column. And we're quite motivated to land in the bottom row. We would like to get a significant result because then we can reject the null hypothesis, say that our research hypothesis was supported, and it's usually more interesting and more exciting and more publishable. So there are a number of incentives working towards pushing us towards that bottom row. And some of the practices we engage in sometimes actually have that effect of pushing results from the top row to the bottom row. And again, researchers are not necessarily and often not aware that that's what the effects of their practice is. So we're just starting to learn about how to make sure we don't accidentally do this. So first I'm going to take you through a caricatured kind of like what the evil scientists would do if they were really trying to push things from the top row to the bottom row, but with the understanding that this is not happening deliberately on the part of researchers. But I want to give you the tools you need to be able to spot when this might be happening without attributing any bad intentions to the researchers who are doing this. So let's take an exaggerated, caricatured example of a researcher who puts a lot of time and effort into a study. They run their study, they get their data, and they probably had a particular research question in mind. Maybe they were interested in whether their mood manipulation, they randomly assigned some people to watch a funny video and some to watch a sad video, and whether that mood manipulation would have some effect on their outcome variable. Maybe their outcome variable is how much you donate to charity. So you get randomly assigned to watch a video that makes you happy or sad, and then you get asked if you're willing to donate some amount to bushfire relief efforts. And so they analyze their data, and the first question they might ask themselves is, did we get an effect of mood on generosity? If they did, then that's a pretty straightforward situation. They can write it up, try to publish it. Journals are likely to be interested in that positive, significant finding. But let's say their first stab at analyzing their data doesn't produce a significant result. So they did not find an effect of mood on generosity when they asked people to donate to a bushfire relief fund. But maybe they threw in some other outcome measures because they were also interested in whether mood affects other kinds of generosity. So maybe they also asked the participants, hey, would you be willing to help us out and just stay an extra 10 minutes and do another study? Because they thought that might be another way to tap into generosity. So you could ask, well, okay, I didn't get the principal effect I was most interested in, but maybe I got some other significant effect somewhere in my data set. I wouldn't want to miss out on that. So let me look around and see if I got that. If I did, I could publish that effect. But the problem is that very often we publish that effect as if that was the one we were looking for all along. That goes by a name called harking, and I'll explain why in a second. Let's say I analyze all my different outcome measures. I still don't get a significant effect. But I'm not done yet. There could still be something significant lurking in my data set. So maybe there's a way to change my analyses, to ask the same questions but a slightly different way, and see if I can reanalyze my data and find a significant effect. So maybe that means I realize after thinking about it that, of course, some of my participants came from University of Melbourne, but some came from Deakin University. And I actually think that Deakin University students are slightly different, so probably I want to control for which university the students came from when I analyze my data. So I add that in as a control variable and see if that changes the result. And if the result becomes significant, now I can say, oh yeah, well, that was the better way to test my question, of course. I'm going to publish that. And if I do that without making it clear that this was not the first thing I tried and that I didn't plan to do that all along, that would be called p-hacking. And we'll get more into detail on that too. And then let's say none of that works. I analyze my data a bunch of different ways, looking at a bunch of different outcome variables, looking at a bunch of different ways to make the statistical model, and none of them produces a statistically significant result. Then I might have no option but to do what we call file drawering the study, which is put it away in my file drawer and not tell the world about it. Partly because journals often aren't willing to use up journal space telling the world about non-significant results, which is a problem. So these three things together, harking, p-hacking, and file drawing, I've kind of put them all under the umbrella of p-hacking. We use that term quite loosely to mean all three. So I'll go back and forth sometimes between using p-hacking to mean just this narrower, reanalyzing your data to get a significant effect, but also it can be used in this broader sense of going from a non-significant result the first time you analyze your data to an outcome that's not ideal, producing a significant result some other way and not declaring how you got it, or ending up with a file drawer full of non-significant results that you don't tell the world about. So let's go into each of these three practices a little bit more. So harking stands for hypothesizing after the results are known. So that acronym spells HARK. And it's a common practice. And in some cases, it happens very much not because of the researcher wanting to do it, but we literally ask researchers to do it. Sometimes journal editors and reviewers tell researchers to rewrite their paper as if they had hypothesized the effect they found all along. So what's happening here is where you design your study, you run your study with a particular hypothesis or research question in mind. You don't get an effect there. So your main goal of your study produces a non-significant effect, but there's some significant effect somewhere else in the data set. So you don't get a significant effect on donations to bushfire relief funds, but you do get a significant effect when you analyze a different outcome variable. And so what you can do is move your target over to what you actually hit. So now you reframe your study to be about that significant effect as if that was the plan all along. And if you keep doing that over and over again, you look like a really good shot. You're like, yep, hit my mark, yep, hit my mark. Now again, it's rare that researchers are intentionally doing this, but what very often happens is that researchers want to make something out of a study. They don't want it to go to waste. They know that journals are only going to be interested in publishing it if they find a significant effect. And they also genuinely believe that there are real effects lurking in there and they don't want to miss them, which could very well be true. The problem is not fishing through your data and finding a significant effect. The problem is that if you do that, you look through your data for a bunch of different effects, and you find one, if you say that was the only effect you were looking for all along. So saying you predicted something when in fact you didn't predict it, you happened upon it by chance, it's very important that the reader knows that that's what happened because it changes our interpretation of the effect. And I won't get into why here, but especially in the context of NHST, the meaning of a p-value changes if it was not the one analysis you planned ahead of time. So harking is not ideal. We shouldn't do it, and it really increases the chances of false positive findings if we do it. It's often taught to students that they ought to do this. It's told, it's encouraged by journal editors and reviewers who ask authors to do it, but it's a bad practice, and it violates the rules of NHST and produces a threat to the statistical validity of our conclusions. What about p-hacking? So p-hacking, in this case, it's the phenomenon of you analyze your data, none of your results are statistically significant, so you try reanalyzing it in different ways, and that has the effect of hacking your p-value until it gets significant. Again, that's not usually what's in the researcher's mind when they're doing it. It's usually not done intentionally. What happens is that we know that there's more than one reasonable way to analyze our data, so we keep analyzing our data different ways, and we want to make sure if there is an effect there that we do detect it. So when we discover something, then we're very tempted to rationalize why that was the right way to analyze our data. And because there is no one true right way to analyze data, that can be a reasonable conclusion to come to, that the analysis that produced the significant effect, that brought the effect out into starkest relief, might be the correct analysis. So some examples are if you keep analyzing your data as you're collecting it, and you check your data along the way, and you stop data collection once your effect is statistically significant, that's a kind of p-hacking. If you have different measures, for example, for your outcome variable, but you only report the effects that reach statistical significance, or there's a bunch of other ways listed on this slide. For example, dropping some participants, maybe after the fact you realize, oh, some participants seemed really sleepy, I'm going to drop the ones that came in really early in the morning, or something like that. So there's a bunch of different reasonable ways you could tweak your analysis that might very well be correct tweaks to make. The problem is when you choose which analysis to report based on which ones produce a significant effect, and when you don't disclose to the readers that you tried other things. So again, the meaning and interpretation of a p-value depends on it having been planned ahead of time. And if you don't disclose that you violated that rule of NHST, then your statistical validity is undermined. And then finally, file drawing. As I mentioned, it's sticking the study in the file drawer, not telling the world about the result. This might be okay sometimes. Of course, some of our studies have irredeemable flaws. We realize after the fact that we didn't administer the questionnaires correctly or something like that. And so those studies maybe don't need to be shared with the world. There's not much to be learned from them. But what becomes problematic is when we file draw our studies just because of the way the result came out, not because of an aspect of the design that we realized there was a flaw, but the result was not what what we wanted or what we think we can tell the world. So if we put those three things together, we have we run a study, we get a messy result. Maybe it's not significant, or maybe it's significant some ways you analyze the data, but not other ways. But we can tidy it up through harking, through p-hacking, and through selectively file drawing some studies. And then we can make a result look much prettier and neater and tidier and more compelling than it really is. So the problem isn't so much with the practices of reanalyzing your data or looking in other places for a significant effect. The problem is in hiding all that messiness and not reporting all the steps you had to go through to get this one beautiful result when actually it's one among many. So let's say a field or a researcher or a team of researchers engages in these practices a lot. What is the consequence for the literature in that field? So if we imagine that the literature were unbiased, that people were not engaging in these practices, were always following the rules of NHST, not p-hacking or harking or file drawing, or being completely transparent about all the steps they had to take, then the literature might have a few very pretty clean results, but it would also have quite a few messy results, and it would likely have many null results. Not all of our research hypotheses are true. Sometimes a null hypothesis is true, and the correct place to end up is a non-significant result. Say we didn't find anything, we weren't able to reject the null. The problem happens when you bring in harking, p-hacking, file drawing, et cetera. You put a lot of incentives on researchers to get pretty results. Then we find ways to turn the messy results into pretty results. We find ways to kind of ignore some of the null results, sweep them under the rug, or not worry about them. And maybe some of the null results we can even turn into significant, pretty results. So what we end up with is a literature with many false positives. Some of these pretty positive results, actually what's underneath should be a null result. That would be the more accurate result. Or in some cases, they're not false positives, but they're exaggerated. The effect size is a lot bigger in the report than it was in the actual data if it was reported in an unbiased way. So we'll talk more about these issues in the next video.